To advance, great. Okay. Well, Susan, thanks so much. And I'm excited to be able to share some of my group's work with you, and particularly with the other speakers today. I'm really pleased to be here. So my job is to tell you a story while talking pretty quickly and hopefully not too loudly to overcome the room next door. But what I'm going to talk about today is work that we've been doing in the laboratory. And these are what I've put here as learning objectives. And I think for this room, there's really no need to emphasize what I have here. I think all of us understand that what we're doing is not only to try to better understand the biology of these cancers that we work on, but because there are things that we cannot offer our patients now that are limitations that are simply unacceptable. So the story I'm going to tell you about today is about glioblastoma. As you guys know, this is a grade 4 primary cancer of the brain. It's the most common cancer that we see in adults. And from my own experience with my patients, these are often people who are otherwise healthy and at the prime of their life. They're people who have young children who are living their best years. The treatments that we offer involve surgical resection, chemotherapy, and radiation. And this is a pretty typical story. This is a patient of mine named Ian Tate. Ian let me share his story and his images. This is what his scan looked like when he presented to hospital in May of 2013. And Ian had the best that we can offer him. He had an awake surgery, we did speech and motor mapping, and he had, as a result, a fantastic resection, went back to work. His wife had a 4-month-old child at the time of his diagnosis. They've since had another child. And for years, his scan looked fantastic until this past summer. And you can see this April 2017 scan, again, was simply, Ian, I'll see you in 3 months. I'm so pleased with how everything looks. And this is what things look like in July. And Ian went back to the operating room in August. And as we feared, what that showed was recurrent tumor. If you look at what we can expect for our patients with glioblastoma, I should be excited that Ian is one of our successes. Median survival for this cancer, despite everything that we do, remains at about 15 months. We've done a better job of extending long-term survivorship to the point where now about 10% of people live for 5 years. But these are not numbers to get particularly excited about. There's a lot more that we need to do. So my laboratory work really stems from the fact that as a clinician, there are limitations that I feel that simply don't allow me to wake up and look at myself in the morning. And the laboratory that I'm lucky to lead really is driven by this question of how do these tumors recur despite what looks like effective therapy? Part of the issue is that GBM is an infiltrative cancer. That's why what I do as a surgeon, no matter how effective it might be, is not curative. If you look at the scan, you can see what looks like tumor. And remarkably, we can do quite a bit to remove this. The difficulty is that if you look at the margin, what you see is this interface between what is solid tumor and adjacent brain and infiltration of tumor cells into the brain. And in fact, if you look up to 2 centimeters away from the solid tumor, our pathologist will still find cells that are cancerous. So we know from the start that surgery, while it may be cytoreductive, while it may reduce the number of cancer cells in the brain, is not going to be curative. The second aspect, and what my talk here will focus on, is this, that glioblastoma recurrence is driven by the fact that there are cells that are capable of resisting the therapies that we have. And I won't spend too much time on this, but there's data from multiple different cancers to show that if you look at cells within these cancer types, they're not all equal. They exist in something that is a hierarchy that is very similar to the hierarchies that we see in normal stem cell systems. In normal stem cells, you have very undifferentiated cells that are highly proliferative and capable of giving rise to progeny, and then more differentiated cells further along. And these cancers appear the same, that there are cells that are like stem cells, and similar to normal stem cells, they have properties that make them resistant to cytotoxic damage. Our stem cells need to last a lifetime. They're remarkably capable of doing that, and these cancer cells co-opt some of those mechanisms to do the same. So we asked, can we disable the defense mechanisms that protect cancer stem cells in glioblastoma from chemotherapy and radiation? And the work I'm going to tell you about today has really been driven by these four folks in my lab, and it's a project that arose from something that we took advantage of from normal system knowledge in neural stem cells. So if you look at neural stem cells, this is from the mouse brain, you have these bona fide stem cells that then can give rise to more differentiated progeny that can amplify and then become either neurons, astrocytes, or oligodendrocytes. If you guys think about what these cancers look like, we see something similar to that in parallel. The work over the last 20 years have really gone towards understanding how this biology in normal stem cells work. And this is work from long ago. Unfortunately, I didn't have hair even then, but what we found back then was that there are signaling pathways that are involved in taking cells that are quiescent, meaning relatively silent, and activating them to become progeny cells. And a lot of this is driven by signaling pathways that are pretty well known and active in normal development. I'll tell you a little story very quickly. We found this agent noggin that impairs something called BMP signaling. And BMP signaling keeps neural stem cells from dividing. When we gave the cells noggin, they divided like crazy. And we actually made this noggin mouse, a mouse that transgenically overexpressed noggin, and it made a remarkable number of neural stem cells. And remarkably, two things happened. One group of mice, they were remarkably smart mice, would, as they got older, start burning out. And when we looked to see why they were burning out, they actually ran out of normal neural stem cells. And it turns out that you need to maintain a small pool in order to have stem cells available throughout life. We called it the Algernon Effect. I don't know if any of you have read the short story Flowers for Algernon, but it was very much like this. We had these brilliant mice that then receded. A second small cohort of these mice developed glioblastoma. And that's what made me think that perhaps there was something about this process of hyperactivating neural stem cells that might be related to the cancers that we saw. I'm going to skip some of this. The cofactor that we found to be the one that really defined this cell that was quiet and that could be activated was something called ID1. And we were very excited by other groups who found that this cofactor ID1 was also a good marker for neural stem cells. One of the things that excited us was that ID1 has been found to be a marker for cancer stem cells in multiple other cancer systems. And it made us ask, could it also be important for glioblastoma? Again, I'm sorry, I'm going to move quickly, but if anyone has something they get stuck on, just put your hand up and I'll stop to answer it. So the first thing we did, which is what all scientists do, we knocked out ID1 and looked to see what happens. So when we knock out ID1 in gliomas, what we find, and you can see a little heat map here, and it doesn't show that great, but what we found was when you inject mice with glioma cells, they all die of glioblastoma. When we injected them with the glioma cells in which we knocked down ID1, the production of those tumors was greatly impaired. And interestingly, this is a survival curve. Those two last curves are mice in whom we've created tumors where we've knocked out ID1. And you can see just doing that one thing about doubles their lifespan. And interestingly, when we looked at them pathologically, the normal mice develop these massive tumors. I don't know if there's a... You can see in the corner there that large tumor in the mouse brain. The mice in which we had put tumor cells that knocked out ID1, we couldn't actually find tumor cells. These mice were dying of hydrocephalus or of buildup of fluid in the brain. And it's not clear to me exactly what happened. It might be that there's an inflammatory response in the brain that actually was able to scavenge the few cancer cells that were there. But whatever the case, what we were seeing was a remarkably different biology just by manipulation of a single gene. This was something I spoke with a friend of mine in England about, and he asked a student of his to do a little examining. And they had this small cohort of patients. As you guys know, for glioblastoma, the general therapy we use is surgery followed by chemotherapy and radiation. And he had a small cohort of patients in whom he had tissue from 3 time points: at their initial surgery, at the time that they had recurrence, and that's pretty typical for us because people often come for a second surgery. But he had a few patients in whom there was a time point in between. For whatever reason, they'd had to go to surgery during chemotherapy and radiation. And because of that, we were able to look to see what the makeup of these tumors was at these 3 time points. And what we found, let's see if I can explain this a little bit. If you look at initial surgery, what you see is that of all the cancer cells, only a small proportion of them are ID1 positive. Most of the cells are ID1 negative. If you look again at recurrence, it's the same thing. Again, most of the cells are ID1 negative. Only a small population are ID1 positive. If you look, though, during chemotherapy and radiation, which is the time when, if you look on an MRI, it looks like there's no cancer there, the cells that are there are all ID1 positive. In other words, there's something about this ID1 positive population. This is the population that's surviving our therapies, and we think sticking around to be able to drive recurrence. So we went in and tested that hypothesis a bit ourselves as well. And what a student of mine in the lab found was that patients who had high ID1 expression after receiving a chemotherapy and radiation had a shorter duration response to both of those therapies. So again, it seems that there's something about this ID1 population that survives therapy and is capable of driving tumor recurrence. We tried to test that a bit in vitro. We know that when we treat cells with temozolomide, which is the chemotherapeutic agent that we use in the clinic, we know that we can kill these cells. This doesn't project well. What we know, though, is that the only cells that survive are the ones that express ID1. Interestingly, when we knocked out ID1, what we could find was that these are cells that were treated with temozolomide or radiation. The top panel is what typically happens. We treat these cells and then we plate them and they form these colonies. When we did the same thing in cells where we had knocked out ID1, they're not able to reform colonies after radiation and chemotherapy. So we took this a bit further and we asked, if that's the case, if we impair ID1, does it make our treatments more effective? So these are cells where we've treated with temozolomide, again, the general therapy for, oh, thank you. How do I do that? This one here.
Green one, yeah, and then the other ones go ahead.
So we've treated these cells with temozolomide, and interestingly, when we treat these cells with temozolomide, they start expressing ID1. This is the cells that express ID1s are the ones that are selected for. When we concomitantly knocked down ID1, we were able to effectively kill more cells. So this is, if you look, this is the baseline number of cells. When we treat with temozolomide, we kill about 40% of those cells. When we additionally inhibit ID1 at the same time, the number of cells that we kill increases tremendously. We try to figure out what is it that allows these ID1 positive cells to survive temozolomide and radiation therapy. And one of the targets that we had was something called EGF. It's a receptor tyrosine kinase that is known to be active in glioma cells, and one of the things that's involved in gliomogenesis. And what we found was that when we knock out ID1, these cells are no longer able to make this growth signal for themselves. And that, in turn, leads to decreased activation of this pathway. This is looking just at phosphorylation of the EGF receptor, but if we look at downstream targets, we saw the same. So it seems, in other words, that chemotherapy kills most of these cells. The cells that survive are the ones that express ID1. And one of the mechanisms by which ID1 affects these cells is by activating EGF signaling and EGFR activation. So this led us to this possibility of wondering, could impairing ID1 in these cells improve the efficacy of the treatments that we already have? As you saw from those pictures that I showed you of Ian's case, our therapies look like they do a relatively good job of controlling these cancers. And what it seems to be the case is that there's a small population of cells that are able to survive that therapy and then likely are involved in recurrence and progression. So it's interesting. I was telling Dr. Malorani back there, I'm on study section right now for CHR, and you read all these grants, and most of us write grants meant for our friends who are also in science. And you get into the depths of trying to answer questions that don't immediately seem like they have resonance for anything that could possibly involve a patient. And this is one of the reasons to have to take the long view. This is work that was driven from publication in a chemical biology journal looking for isotopes that impair something called USP1. And I'll tell you, looking back at the initial paper, I have no idea why this group was interested in finding a chemical inhibitor of USP1. But whatever the case, someone picked up on it and realized that USP1 is a mediator for osteosarcoma and that this chemical inhibitor could be important for patients with that disease. And it turns out that the way that this works is that it impairs a moiety that's also shared by ID1. And the interesting thing is that the chemical biology group that created this small molecule that impairs that moiety said, well, look, it turns out that there's actually a drug on the market that's an even stronger binding agent for that moiety than what we've designed. And it's this. It's a drug called pimozide. So pimozide is an antipsychotic agent. It's a drug that we use to treat schizophrenia. It is the second most commonly prescribed drug in Canada for schizophrenia. It readily crosses the blood-brain barrier. It is a generic drug and cheap as heck. My student asked, could we impair ID1 using this drug in the same way that we have using SI and shRNA? And what you can see here, this is ID1 expression. So typically, again, when you look at the majority of these glioma cells, the population that expresses ID1 is quite small. And that population only becomes readily visible once you kill most of the cells with temozolomide. The cells that are left are the ones that express ID1. And what we found was that if we concurrently treated these cells with pimozide, you don't enrich for that population. Putting that another way, what we saw is that treating with pimozide sensitized these cells to cell death because of temozolomide. And we saw the same with radiation therapy. So it's very nice to be able to do this in a dish. It's a much harder thing to do with tumors that are in the brain. So that was our next step, to try what would happen if we did this with tumors in the brain. We started first with a flank model. So this is pretty far from the human disease. This is taking a glioma cell line and putting it in the flank of an immunocompromised mouse. We do this because it's easy. You can take a little caliper and look to see if your tumor's growing. And what we did was treated these patients of ours with pimozide for a week before we gave them temozolomide, and then continued the pimozide therapy for a week thereafter. And this is what we saw. So when you give these mice temozolomide, this is before we start treatment. What you can see is that these unfortunate mice have tumor that have grown in both flanks. And temozolomide looks like it works. You give them temozolomide, and the tumors regress. We only took this out to 30 days. If you take it out to 45 days, what you see is that the mice almost can't walk because the tumors are so massive. When we did the same with temozolomide and pimozide at the same time, the initial response doesn't look to be that significantly different. But if you follow them out now, and I'm sorry somehow this dropped off to 45 days, what you see is that this is still all that we have. And this is what things look like at 45 days. This is treating with, this is looking just at tumor weight. These are our control mice. These are the tumors weigh almost as much as the mice do at 45 days. This is what they look like with temozolomide. So it's not that the temozolomide doesn't work, but it only holds these tumors off for so long. And this is what it looks like with Pemazide. So again, all very good to do this in the flank of a mouse. And the next question is to ask whether or not we see the same effect when these are tumors that are within the brain itself. So we did something very similar. Again, these mice had tumor cells injected into the striatum. We treated these mice with Pemazide for a week. The Pemazide therapy continues for 3 more weeks thereafter, and during week 2, they received temozolomide. And this is what we see. It's encouraging, not as encouraging as I'd like, but if you look at these mice that receive temozolomide, again, the tumor responds to therapy. By 1 month, tumor is disseminated throughout. This is what we gain with Pemazide. It's about a 10-day extension of life. Statistically, it's profound. I'll say, sadly, this will get me a fantastic paper. What it tells us is that we've disabled one mechanism by which these cells are resistant, and it's very far from being the whole. And that's what we're studying now. What we have seen is that these cells are able to activate other ID proteins that can compensate for this one and that aren't blocked by Pemazide. What this has allowed us to say, though, is that we know that there are reservoirs, small reservoirs of cells that only make up a small part of the tumor at presentation that are responsible for progression here. What we know is that we're able to target those partially. The targeting that we do seems to improve the efficacy of the chemotherapy agents that we use, and it's very far from being a complete answer. So, if you think about what a 10% extension of life in our patients could mean in the clinic, using a relatively inexpensive drug that has very few side effects, it's something that is, in my mind, worth trying. It's as much as we get with second-line chemotherapeutic agents that we use right now. My hope is as well, and we're doing work on this now, is we talk about glioblastoma as a disease. And in medicine, we talk about diseases and syndromes. Syndromes are clinical phenomena that differ scientifically or differ biologically. And the interesting thing about glioblastoma, and this is the case for many cancers, is that glioblastoma is something that we've defined because it looks homogeneous under a microscope. And the more that we look at it biologically, the more it's starting to look like a remarkably diverse set of diseases. My hypothesis is that there are likely subsets of glioblastoma in which ID1 positive populations are the main drivers of stemness. And those are the ones that would be appropriately targeted by a therapy like this. This is a question we're trying to answer right now. What is it in the general glioblastoma population? If we've knocked out this small subset of cells, what is it that allows these tumors still to recur? So I'll leave with this. Glioblastoma right now is an incurable malignant brain cancer that affects young, otherwise healthy people. Our current therapies are simply inadequate, which makes me excited to see as many students in the room as I do. Cancer recurrence in glioblastoma is driven by cells that are resistant to the therapies that we have and that I'd argue are cancer stem cells. And targeting the processes that maintain those cancer stem cells in glioblastoma can improve upon our current therapies. So this is Ian. This is his daughter, Jenny, who was born, you can see, about 1.5 years ago now. This is why we get together like this. This is why we do the work that we do. It's professionally the fact that I need to win grants, that I need to have money to do the work that I do, to have the students that I have. But this is it. And somehow we have to keep our eye on what it is that drives us. So I'll say again, none of this is possible without the talent of the people who I am lucky to have working with me, and with the support from the multiple different groups that I've gotten. Everything that we do is so expensive and so time demanding, and becoming even more so. And hopefully you'll see from just a little bit of what I showed you today, we're getting closer to being able to actually deliver on some of the things that we are trying to work on. I owe all these people thanks, and I'll say the CPTF supported a student of mine this summer in the lab who did fantastic work, and we can't do it without that sort of support. Thank you.